Good morning, everyone. Today, my topic of journal club is Hallmarks of Cancer. These two researchers you're seeing on the screen, on the left side is Robert A. Winberg, and on the right side is Douglas Hannan. They have done tremendous research works in the cancer field, and one of their landmark paper, which was published in 2000, was the Hallmarks of Cancer. So in 2000, they gave us 6 core hallmarks because at that time there was no unified model of cancer, and cancer was just and merely an uncontrolled cell proliferation. Uh, there was a somatic mutation theory, but it was not fully explained. Pathways were known which causes cancer, but were not unified, and the cancer was specifically seen as merely a simple cell which invades and metastasizes. But according to them, the cancer cell biology was more complex, including the different types of cells which helps cancer to invade, proliferate, and metastasize. So if we talk about the carcinogenesis, when a normal cell first get a carcinogen induced mutation, which we call as initiating mutation, it further uh, acquires the stem cell like properties, which further gets additional driver mutations and acquire these cancer hallmarks to form the founding cancer cell. And that cancer cell again gets additional mutations, uh, further gets the genetic evolved and leads to and becomes a genetically heterogeneous cancer. Into answer.
Before that, I'm just to tell you one more thing about this breast cancer. There was a hypothesis put forward by Bernard Fisher, very famous thing, that cancer breast to start with is systemic. So whatever local treatment we are doing for this, we are really going to cure the fever. So there was a common thing put forward that local control does not mean cure, but cure does not achieve without local. So this is these slides which we are going to present. They are very important so that it will highlight the pathophysiology of the cancer and what happens in a single cell and mutation occurs and carcinoma.
And in so, whenever first a normal cell gets a carcinogen induced mutation, which we call as initiating mutation, that normal cell acquires a stem cell like properties, which further gets an additional driver mutations, and those mutations acquire the cancer hallmarks, forming a founding cancer cell. And that founding cancer cell further grows on additional mutations, further gets genetically evolved into a genetically heterogeneous cancer. In 2000, they gave a emergent circuit of the cell, believing that a cancer cell has a big network regulating circuits that governs the normal cell proliferation and homeostasis. So according to them, cancer is a disease caused by dynamic genetic changes that transform normal cells into malignant ones through multiple steps. And these are driven by the oncogenes, tumor suppressor genes. Due to the cancer's complexity, research shows that a few core principles and cellular traits underlie most cancers. And these discoveries point to common mechanisms regulating cell growth, death, and interaction with the environment, which cancer disrupts to develop and progress. So, due to this complexity, there were a number of questions which were raised. So, how many distinct regulatory circuits within each type of target cell must be disrupted in order for such a cell to become cancerous? Does the same set of cellular regulatory circuits suffer disruption in the cells of the disparate neoplasm arising in the human body? Or which of these circuits operate on a cell autonomous basis and which are coupled to the signals that cell receive from their surrounding microenvironment within a tissue? So in 2000, they gave this emergent circuit, and they basically in next paper in 2011 they named those circuits. They gave different names such as motility circuits, differentiating circuits, viability circuits, proliferation circuits, and eventually all these circuits lead to changes in the gene expression and causing cancer. Another thing which they highlighted in these papers was that tumor microenvironment. A tumor microenvironment is increasingly recognized as a complex, dynamic organ-like system composed of cancer cells and a variety of the supporting cells. We know tumor is basically made of stromal cells and parenchymal cells. Parenchymal cells are the cells which are cancer cells, cancer stem cells, and then the stromal cells are the cells which basically supports these cancer cells to grow, invade, and metastasize. And these stromal cells are endothelial cells, pericytes, uh, immune inflammatory cells. And there is a big connection between these both cells and regulatory network which helps them to metastasize and invade further. Before going to the hallmarks, I just want to explain a little bit about the proliferation. As we can see on the screen, we have cell cycle. So we know that cell cycle has metaphase, as mitotic phase, and then interphase. In interphase, we have G1, S, and G2. And if the most important two phases of the cell cycle are basically the mitotic phase and the synthesis phase, the S phase. And any mutation or defect which is carried towards these phases can lead to the cancer. There's different cell cycle components which helps a cell cycle to proliferate or which helps the cell cycle to decide whether the cell should proliferate or not. If I talk about these two checkpoints written on the screen, the G1S checkpoint and G2M checkpoint, these are the really important restriction points which decides whether the cell should progress further or not. And G1S checkpoint is really, really important because once the mutation goes forward from this G1S checkpoint, that cell will undergo, that will be a complete cell cycle. So if mutation pass from the G1S checkpoint, that will be carried on further towards the mitosis and the cancer can happen. So we have cell cycle components such as cyclin and cyclin dependent kinase, which basically allows the cell to progress through this G1 checkpoint. Then we have those cell cycle inhibitors which inhibit these cyclins such as CYP family and INK4 family. And then we have two really important checkpoint components, which is RB and P53, which are tumor suppressor genes. So conceptually, there are three types of cancer genes. So we have oncogenes, we have tumor suppressor genes, and then we have DNA repair genes. And in normal cell, the balance between this proto-oncogene and tumor suppressor gene is there. So if a proto-oncogene gets mutated to the oncogene, that means there will be too much proliferation. So oncogene is basically a gas, it means too much proliferation, and the tumor suppressor gene is basically brakes, means which controls the proliferation. So too much oncogene, too much gas means leading to the cancer, and whereas if there's no brake, tumor suppressor gene is inactivated, it will eventually lead to cancer. And this is the table taken from the Robbins Book of Pathology, where they have mentioned different oncogenes. So basically we have proto-oncogenes, which further gets overexpressed or amplified or get mutated into oncogenes and cause different associated human tumors. For example, we have proteins involved in the signal transduction. As we can see, GTP binding proteins such as KRAS, HRAS, NRAS. Whenever there's a point mutation in any of them, they eventually lead to the cancer. We have nuclear regulatory pathways such as transcriptional activators such as MYC and NMYC, which again have translocation or amplification can lead to further types of cancers. If I talk about tumor suppressor genes, as we can see on the screen, we have different genes having different functions, and if they get mutated, can lead to familial syndromes and sporadic cancers. Two really great examples, RB and P53. RB is written in there in the left side. It's an inhibitor of cell cycle progression. So the function of RB is basically it's inhibit the G1S transition during the cell cycle progression, and if it's mutated, it can lead to familial syndromes or sporadic cancers. On the right side, there's TP53 under the enablers of genomic stability, and TP53 function is basically to sense any kind of mutation or defect so that it can allow the apoptosis in response to the DNA damage. And mutation in T53 can further cause syndromes and cancers. Another is the DNA repair genes such as BRCA1, BRCA2, which helps us in repair of double-stranded breaks in DNA. And any mutation in these genes, as we know, can lead to familial breast and ovarian cancers. Coming back to this specific part of the cell cycle, uh, we have different growth factors which allow cells to proliferate, and then we have growth inhibitors which stops the cell to not proliferate. So basically, the RB is actually bound to elongation 2 factor, and whenever we need the proliferation, this elongation 2 factor is released and is bound to the site, transcription site, and the transcription is activated, and the cell further goes from G1 phase to the S phase. So let's say for growth factors, whenever growth factors bind to the growth factor receptors, the cyclin DNA gets activated, they hyperphosphorylate the retinoblastoma, and the elongation 2 factor is released, it's bound to the site, and the transcription happens. On the other side, if I talk about growth inhibitors, they actually stimulate these CDK inhibitors, the CYP family and INK4 family. They inactivate those cyclins, so the RB remain hypophosphorylated, so the transcription is. Another really important uh, in the cell cycle progression is the downstream signaling pathways. So these are the pathways which are activated after the growth receptor stimulation, which drives the cell cycle progression. And two really important pathways which are also really mentioned again and again in the hallmarks is these two pathways. One is the MAPK pathway shown on the left side, and then we have PI3K mTOR pathway which is on the right side. So the MAPK pathway is basically whenever any epidermal growth factor binds to the receptor, they activate the RAS, and that RAS, activated RAS, further activates the RAF, MEK, and ERG, and leads to the uh, proliferation. On the other hand, the PI3K pathway, whenever PI3K get activated, it converts the phosphorylated 2 to 3, and then further PIP3 activates AKT, which also has a function of survival, increase the survival in the cells, and again it activates mTOR, which further help in the proliferation and angiogenesis. So coming back to the hallmarks, in 2000, on the left side of the screen, you can see the 6 core hallmarks which were given in 2000 by the Winberg and Hanan. So these were self-sufficiency in growth signals, evading apoptosis, insensitivity to anti-growth signals, sustained angiogenesis, tissue invasion and metastasis, limitless replicative potential. In 2011, they uh, they named these 6 core hallmarks, and they also gave additional 2 emerging hallmarks which were deregulating cellular energetics, avoiding immune destruction, and they gave 2 enabling characteristics, that was genome instability and mutation, genome instability and mutation, and tumor promoting inflammation. So hallmarks is something which helps the cancer cell to acquire the mutation and to become the uh, which becomes helps it to become malignant. And the enabling characteristic is something which facilitate those hallmarks, or which facilitate the cancer cell to acquire those hallmarks fastly. And later in 2022, they included these four hallmarks with those 6 core hallmarks and given 10 core hallmarks. So now we have 10 core hallmarks and 2 emerging hallmarks which are written in blue, which is unlocking phenotypic plasticity and senescent cells, and 2 enabling characteristics, non-mutational epigenetic reprogramming and polymorphic microbiomes. So starting from the first hallmark is sustaining proliferative signaling. So when we talk about normal cell, the normal cell depends on signals from outside the cells, such as growth factors, cytokines, extracellular matrix, and cell-to-cell adhesion molecules. And when these signals are received by specific transmembrane receptors in the cell surface, it leads to cell growth and proliferation. But cancer cells escape this control and start growing without even needing those external signals with following mechanism. So we have autocrine and paracrine stimulation. So when cancer cells overproduce growth factors by themselves effectively, stimulating their own receptors continuously without even any external input, which is known as autocrine signaling. And they also coerce the surrounding stromal cells to release the growth factors that fuel the cancer growth. So cancer cells may increase the number of growth factor receptors in their surface or acquire the mutations that make this receptor active without the presence of the ligands. And mutations can also activate signaling molecule downstream of the receptors, bypassing the need for receptor altogether. Another mechanism is somatic mutations and constitutive signaling. Various genomic studies of tumor have identified the mutations that causes continuous activation of signaling pathways typically triggered by growth factor receptor such as BRAF. So, as BRAF mutation in melanoma, we know about 40% of the melanomas have mutation in BRAF, which is a component of the MAPK pathway, causing constant proliferative signals. And these mutations allow the cancer cells to grow independently of the normal external signals, and different pathways can be affected, influencing the tumor behavior and response to therapies. Another mechanism is disruption of the negative feedback mechanism. So cells have built-in negative feedback loops that keep proliferative signals check and maintain homeostasis. One of these mechanisms are written there's RAS mutations. So again, the two downstream pathways which I have explained earlier, this is the same pathways. So we have the MAPK pathway and we have the PI3K pathway. So if whenever we need proliferation, the growth factor bound to growth factor receptors, the inactive RAS gets active and further activate the transcription. But if we have mutation in the RAS, so the active RAS normally, whenever we do not need proliferation, that active RAS gets inactivated by hydrolysis. And when there's a mutation, that active RAS cannot get inactive and it stays active. And that active RAS always leads to the cellular proliferation. Another example is loss of PTEN. So PI3K, as I earlier mentioned, that PI3K converts phosphoinositide 2 to 3, which further activates AKT. Now, the function of PTEN is to convert back that phosphoinositide 3 to phosphoinositide 2. So if we have loss of PTEN, there will be continuous activation of AKT leading to the cell death. Another hallmark of cancer is evading growth suppressors. So cancer cells not only promote their own growth signals but also actively evade mechanisms that normally suppress proliferation. And this ability of evading the growth suppressors is critical for uncontrolled tumor growth and involves disrupting the tumor suppressor genes. So role of tumor suppressor genes is basically acting as a brakes on the cellular proliferation. Whenever there's an unfavorable conditions such as DNA damage or inadequate nutrient or stress signals, they halt the cell cycle. And two most important well-defined tumor suppressors are RB, retinoblastoma protein, and TP53. So RB acts as a gatekeeper for the cell cycle. It integrates multiple internal and external signals to decide whether a cell should proceed to divide or pause. And normally, RB restricts the cell cycle progression from the G1 to the S phase. So loss of RB function removes the critical checkpoint leading to the uncontrolled proliferation. About TP53, often called as the guardian of genome, it basically functions as monitoring the internal cellular stress, such as DNA damage, nutrient deprivation, or hypoxia. So it can pause the cell cycle to allow repair or induce cellular senescence or apoptosis if damage is irreparable. But there's a thing called network redundancy and its implications. Although the retinoblastoma and TP53 are key regulators, the cellular proliferation control network is functionally redundant. Evidence shows that the loss of RB or TP53 alone in the mice often does not immediately cause rampant proliferation of the cancer. For example, mice with cells lacking the RB throughout many tissues often still shows the normal tissue morphology and only develops tumor late in specific organs. Similarly, in TP53, null mice also develop normal with proper tissue homeostasis before cancer arises later. So this suggests that there are other mechanisms that compensate for the loss of these suppressors, limiting immediate tumor genesis but potentially falling over time or in combination with the additional mutations. Another mechanism is by contact inhibition and evasion. So a normal cell, whenever so a distinct form of growth suppression occurs when cells densely contact each other. That means whenever a normal cell grows and it feels it has achieved the size, it will stop proliferating. This contact inhibition stops normal cell from proliferating excessively, forming organized tissue structures. And cancer cells frequently lose this property, allowing continued division even in the crowded environments. Two tumor suppressors which are involved in the contact inhibition are Merlin and LKB1. Merlin, we know, is a links with the cell adhesion molecules and E-cadherin. So basically, it links E-cadherin to the receptor tyrosine kinase, such as the EGF receptor, and basically strengthens the cell-to-cell adhesion and sequester growth factor receptors. If there's a disruption of Merlin, as we see in neurofibromatosis type 2, this disrupts contact inhibition and actually supports the tumor development. Another one is LKB1, which we found that LKB1 mutations are found in some human cancer, highlighting its role as a growth suppressor. The main function of the LKB1 is it maintains the epithelial tissue architecture and this suppresses the transformation by oncogenes such as MSC. Another pathway is TGF beta pathway from suppressor to promoter of malignancy. So transforming growth factor beta is a cytokine that normally inhibits the cellular proliferation and function as a tumor suppressor in early stages of the cancer. Cancer cells finds a way to evolve and ways to evade the TGF beta suppressive effects, including losing the responsiveness to the TGF beta anti-proliferative signaling, rewiring the TGF beta signaling to promote the epithelial to mesenchymal transition, and the switch from a tumor suppressive to pro-metastatic role exemplifies how cancer cells manipulate growth suppressor pathways to their advantage. Next hallmark is resisting cell death. So normally, programmed cell death process like apoptosis, which acts as a natural barrier. Whenever there's a damage, we have mechanisms in our body which helps us to destroy the cell, such as apoptosis and necrosis. So apoptosis is basically programmed cell death. So in the right side of the screen, we can see an image in where intrinsic pathway of apoptosis has been shown. And apoptosis actually works with the help of p53. So apoptosis also have different genes, different proteins. So we have pro-apoptotic proteins, we have anti-apoptotic, and then we have the sensor genes. So pro-apoptotic will be helpful in proliferation, anti-apoptotic will be helpful in stopping the regulating those proliferation. So whenever a normal cell has to proliferate, so whenever there's a DNA damage, the p53 will check for that damage and will activate those sensors such as BAX and BAC, and then it will lead further to the apoptosis. But if there's a mutation of any p53, or there's a loss of p53 function, that will disable the key sensors and it will prevent the apoptosis. So cancer cells develop various mechanisms to suppress the apoptosis. So first is the loss of p53 function. Second is upregulation of the anti-apoptotic proteins such as BCL, MCL. And another one is downregulation of the pro-apoptotic proteins. So lower level of BAX, PIM, or sensor proteins like PUMA reduce the cell ability to undergo apoptosis. Another one is impairment of the extrinsic death signaling. So the cancer cell can block the death receptor activation pathways. Another mechanism is autophagy. The autophagy is another form of programmed cell response where cells degrade and recycle their own components during stress, especially nutrient deprivation. So autophagy involves the formation of autophagosomes that engulf cellular organelles and fuse with lysosome for degradation. This regulation overlaps with the apoptotic pathways, and usually a protein known as Plaquenil-1 protein is essential for autophagy and is connected to the apoptotic BCL2 family proteins via BH3 domain. So if there's a genetic loss of the Plaquenil-1 or autophagy, this gene increases cancer susceptibility, indicating that autophagy can act as a barrier to tumorigenesis. However, autophagy can also promote tumor cell survival by helping the cancer cells endure the metabolic stress and survive treatment. So some cancer cells use autophagy to enter a dormant drug-resistant state, contributing to the tumor relapse. Another factor is necrosis. As unlike apoptosis, so necrosis and apoptosis are basically leading to the cell death. The main difference is that whenever necrosis happens, it also has an inflammation. So whenever necrosis happens, the cell swells, cell ruptures, and release that cellular contents in the tissue environment. That means if the necrotic cell triggers a death, the inflammation will be released by releasing the pro-inflammatory molecules and recruiting the immune cells. Although inflammation normally helps clear dead cells in the tumors, but inflammatory cells can paradoxically promote cancer by enhancing the blood vessel formation, stimulating the cancer cell proliferation and invasion, and also necrotic cells release factors like interleukin 1 alpha, which can directly encourage proliferation of nearby tumor cells. Thus, while necrosis eliminates the damaged cells, it can inadvertently support tumor growth and progression by creating a pro-tumor microenvironment. Next hallmark is enabling replicative immortality. By name suggests itself, is the ability to replicate indefinitely. So it's a really crucial property cancer cells must acquire to form large clinically evident tumors. And there are some barriers for the unlimited replication. One is senescence and crisis, the mitosis crisis. So normal cells typically face two main proliferation barriers. One is senescence, a state in which cell permanently stops dividing but remains metabolic active. This is usually irreversible. Another is mitotic crisis, a phase characterized by widespread cell death, primarily due to genetic instability caused by chromosome abnormalities. So when normal cells are cultured, they usually divide only a limited number of times before entering senescence, and some cells may bypass senescence but ultimately enter crisis, where most of them die. Rarely, a cell escapes that crisis and gains the ability to proliferate indefinitely, and this process is known as immortalization. There's a big role of telomeres and telomerase. So what telomeres is, they are the repeated DNA sequences at the end of chromosomes to protect the chromosome integrity. And every time the normal cell replicates or normal cell divides, the telomeres get shortened. So telomeres shorten progressively with each cell division, eventually causing loss of chromosome and protection, formation of unstable dicentric chromosome leading to chromosome rearrangements and dysfunction, and then entry into crisis or senescence once the telomeres become critically short. On the other hand, we have telomerase, which basically adds back the telomeres onto the chromosome ends. And normally, in normal cells, the activity of telomerase is really less or very low, but in the cancer cells, it is expressed really high. So what telomerase does is, by extending the telomeres, they prevent the critical telomere shortening. They enable cells to bypass the senescence and crisis, and they confers unlimited replicative potential immortality. This can be explained by this picture. So as we can see, when the telomerase is inactive, means telomeres are keep on getting shortened with the multiple cell divisions. Now, when the cell has achieved those cell divisions, the p53 will be activated, and the cell will undergoes the senescence. If there's a mutation in p53, the cell will pass that G1S checkpoint and further goes to the mitosis. Now, in mitosis, there's a way of again checking the mutations by the help of the non-homologous end joining pathway. So when this mutation goes further into the mitosis, there's a bridge fusion breakage cycle where it keeps on getting new double-stranded breaks all over again and again. Now again, if there is telomerase absent, which is happening normally, that will lead to the mitotic catastrophe. But if telomerase is reactivated, as we see in the cancer cells, it will further lead to the cancer. Next hallmark is inducing angiogenesis. So tumors, like normal tissue, needs a steady supply of oxygen and nutrients and an efficient system to remove waste products. And this critical support comes from the formation of new blood vessels, a process called angiogenesis. And this is regulated by something known as angiogenic switch. So angiogenic switch is controlled by a balance of factors that promote or inhibit the blood vessel formation. We have pro-angiogenic factors, for example, vascular endothelial growth factor a key protein that stimulates the endothelial cells to grow new vessels. And we have anti-angiogenic factors, for example, thrombospondin 1, which binds receptors on endothelial cell to suppress the angiogenesis. So multiple molecular layers modulate these signals. So VGF ligands can be stored in the extracellular matrix and released by protease, enabling precise local activation of the angiogenesis. And other growth factors such as fibroblast growth factors family also contribute to tumor blood vessel formation. Tumor cells can directly induce the angiogenesis through oncogene activation, such as RAS and MYC, upregulating those pro-angiogenic factors, and immune cells infiltrating tumor cells can also produce these signals, underlying how distinct cancer hallmark traits such as sustained proliferation and angiogenesis can be co-regulated. The body produces endogenous inhibitors that modulate the angiogenesis to maintain tissue balance and restrict that inappropriate vessel growth. Beside the thrombospondin 1, other inhibitors are angiostatin and endostatin. And these inhibitors circulate normally and prevent the unchecked angiogenesis, but are often overridden in the cancer. And mouse models have shown that loss of such inhibitors enhance tumor growth while increasing their level impairs tumor progression. Next hallmark is activating invasion and metastasis. So this process by which cancer cell invades surrounding tissue and spread to the distant sites. So it happens through activating invasion and metastasis. So progress over recent decades has shed light on the complex cellular changes, molecular pathways, and interaction that enable these capabilities. So first mechanism is cell adhesion and invasion. So when there's a loss of E-cadherin, so basically E-cadherin is a cell-to-cell adhesion molecule that maintains the structure. And loss or downregulation of this E-cadherin disrupt tissue architecture, enabling increased motility, detachment of cancer cells from their neighbors, enhanced inhibitory, and this is supported by frequent downregulation or mutation of E-cadherin in many human carcinomas. We have invasion metastasis cascade. So we have different steps. So first we have local invasion. So cancer cell infiltrates in the surrounding tissue. We have intravasation, entry into nearby blood or lymphatic vessels. Then we have transit, circulation through lymphatic and blood vessels. Extravasation, now they exit from the vessel into a distant tissue parenchyma. Micrometastasis formation and colonization. And each step demands specific cellular adaptations. Two more mechanisms which helps cancer to metastasize is epithelial mesenchymal transition and also stromal cells. Now stromal cells I have explained earlier in that picture in the tumor microenvironment that stromal cells supports the cancer cells to further disseminate, invade, and metastasize. When we talk about metastasis, it occurs in two main phases. We have dissemination and colonization. The dissemination is basically physical spread of cancer cells from the primary tumor, and colonization is adaptation and growth in distant organs. But many micrometastases disseminate but fails to grow into overt tumor, potentially due to dormancy induced by lack of angiogenesis, nutrient deprivation, anti-growth signals, and immune system suppression. So if I explain this picture, we can clearly see the metastatic dissemination happening. So there's a connection between the parenchyma and the stromal cells. So whenever we have the cancer cells, the incipient cancer cells or the cancer stem cells, they recruit the stromal cells to the lesion. So now there's a relation going on between the parenchyma and stroma. And whenever the stromal cells undergo primary tumorigenesis, they form nascent stroma of the primary tumor, which further helps the incipient cancer cells to form the further cancer cells and cancer stem cells, which further helps the nascent stroma to form the supportive stroma of the primary tumor. And that supportive stroma of the primary tumor helps that cancer cells to form invasive cancer cells. And these invasive cancer cells now can further disseminate and cause micrometastasis. And when these micrometastasis happens, they recruit the stromal cell to the seeded metastasis and forming the normal stroma and including the normal stroma. Now this normal stroma again becomes nascent stroma of metastasis, again leading to the macrometastasis and supporting stroma of the metastasis happening. So basically this actually explains the seed and soil theory. So the cancer cells and cancer stem cells are the seeds and the normal stroma or the stroma acquired by these cancer cells are the soil. So whenever the cancer goes to a specific space or place, they make an environment for themselves before even metastasizing or after going there. Another hallmark is genomic instability and mutation. So cancer progression depends heavily on genetic alterations And one of the key enabling factors underlying these changes is genomic instability and mutation. And this instability accelerates the accumulation of mutations and epigenetic changes that grant the cancer cell selective growth advantages, where clonal evolution and mutation acquisition, so they acquire either genetic mutations or they undergo epigenetic alterations. So cancer cells often display elevated mutation rates due to increased exposure or sensitivity to the mutation factors, and the gene involves are DNA damage detection or repair pathway activation, such as ATM or ADR, direct DNA repair mechanism, example BRCA1 or 2 in homologous recombination, and neutralizing those mutagenic response. And loss of these caretaker functions leads to accumulation of mutation and genomic aberrations that fuel the tumorigenesis. Next hallmark is tumor promoting inflammation. So tumor promoting inflammation is a key enabling characteristic in cancer development and progression. While inflammation is typically a protective response to injury or infection, paradoxically it fosters the tumor growth and malignancy. So inflammation contributes to multiple hallmark capabilities of cancer by supplying the bioactive molecules to the tumor microenvironment. So inflammatory cells such as TAMs, cytokines, chemokines, and growth factors that stimulate the cancer cell proliferation, survival, angiogenesis, and invasions. So there's an early role in tumor initiation and progression. So inflammation also is evident in the earliest stage of neoplastic progression, and the key signaling pathways involved in that tumor promoting inflammation includes nuclear factor kappa beta, JAK-STAT, MAPK. Whenever these pathways are activated by the cytokines and other inflammatory mediators, regulating gene involved in cell survival proliferation, immune evasion, and EMT. Next hallmark is deprogramming energy metabolism. So as we know, the cancer cells reprogram their energy metabolism to support the uncontrolled proliferation, a hallmark of the neoplastic disease. So basically, normal cell in our body converts the glucose to pyruvate and basically uses the oxidative phosphorylation, efficiently generating the ATP. But in anaerobic condition, the cell rely on glycolysis, giving less ATP but maintains energy production. But the cancer cells, however, preferentially metabolize glucose via glycolysis even when the oxygen is abundant, hence naming aerobic glycolysis. So even this glycolysis, the aerobic glycolysis gives less ATP. The cancer cell actually compensates by increasing the glucose uptake via the GLUT1 transporters. They upregulate the glycolytic enzymes. They have hypoxic tumor microenvironments which further induce the glycolysis through stabilization of the transcription factors. And this preference for glycolysis despite lower ATP supports rapid cancer cell growth by diverting the glycolytic intermediates into biosynthetic pathways vital for proliferation. So these include the nucleotide synthesis, amino acid synthesis, and lipid synthesis for the membrane. And such biosynthetic demands are critical for producing the macromolecules and organelles in dividing cells. Next is evading immune destruction. So evading the immune destruction is also a critical hallmark for cancer, allowing tumor cells to escape the detection and elimination by the immune system. So normally the immune system monitors tissue and destroys the abnormal cells, including early cancer cells, through a process called immune surveillance. So we have two processes. We have immune surveillance and immune editing. Immune surveillance is basically cytotoxic T cells, and natural killer cells constantly patrolling the body for abnormal cells and eliminating them. Evidence from the animal studies showed that mice with a weakened immune system develops tumors more frequently and rapidly, highlighting the immune system role in suppressing cancer. Another process, immunoediting, where the immune system shapes tumor evolution by eliminating highly immunogenic cancer cells, leaving behind the weakly immunogenic variants. So basically, how cancer cell evades the immune destruction, they downregulate or lose major histocompatibility complex 1 molecules, making them less visible to the T cells. They secrete immunosuppressive factors like TGF-beta, which inhibit the activity of cytotoxic T cells and NK cells. Cancer cells recruit regulatory T cells and MDSC, which actively suppress the immune response, and also the immune checkpoint activation, such as cancer cell upregulate the immune checkpoint molecules. So next is the emerging hallmark, which is unlocking phenotypic plasticity. So basically, phenotypic plasticity includes three main processes. We have dedifferentiation, block dedifferentiation, and trans dedifferentiation. So when we talk about the normal differentiation, the progenitor cells normally divides and normally forms a cell. In dedifferentiation, what happens is that cells normally undergo differentiation, forms a cell, but then further go back to the progenitor state. So divides normally and go back again to the progenitor state. In block dedifferentiation, what happens is the cell cycle progression halts in the midway and forms a lot of immature cells, as we seen in the leukemia. And in trans dedifferentiation, what happens is the cancer cell actually changes the lineage in between. So a cancer cell, for example, in the basal cell carcinoma, can switch to a different cell type to escape the hedgehog pathway inhibition, acquiring resistance to the target therapy. So phenotypic plasticity is really important in therapeutic resistance because phenotypic plasticity is also a major factor as it can switch lineage or states to evade the targeted treatment. Next is the enabling characteristics such as non-mutational epigenetic reprogramming. So we know that there is mutational epigenetic reprogramming, but non-mutational epigenetic reprogramming is also an essential enabler characteristics, which operates independently of the DNA mutations. So epigenetic reprogramming mechanisms such as DNA methylation, histone modification, and chromatin remodeling regulate gene expression and are central to normal development and differentiation. In cancer, these mechanisms are hijacked to drive hallmark capabilities, and the tumor microenvironment significantly influences the epigenetic reprogramming. So factors like hypoxia, nutrient deprivation, physical properties of extracellular can induce more epigenetic changes. So non-mutational epigenetic reprogramming is a bona fide enabling characteristic that facilitate the acquisition of the hallmark capabilities in the cancer. Next, enabling characteristics is polymorphic microbiomes. So as we know, every barrier in our body has different microbiomes. So these different barriers modulate these different barriers have different microbiomes which modulate different tumor growth, inflammation, immune invasions, genome instability, and therapeutic resistance. So microbiome actually modulates both innate and adaptive immune response, and bacteria can reach the gut barrier, trigger immune responses, create tumor promoting or tumor antagonizing immune microenvironments. And also there's an intratumoral microbiota. So bacteria have been detected within the solid tumors and their presence is associated with clinical pathological features. The studies shown that the tumor microbiome can influence tumor promoting inflammation and malignant progression. The last hallmark is the senescent cells. As it was earlier discussed in one of the hallmarks, now, cellular senescence is considered as the main hallmark, the emerging hallmark. So cellular senescence is a state of irreversible cell cycle arrest that acts as a protective mechanism to maintain the tissue homeostasis by inactivating and removing damaged, dysfunctional, or unnecessary cells. This process is triggered by various stresses, including DNA damage, nutrient deprivation, and signal imbalance, and is a key feature of aging and cancer biology. So there's dual role of the senescence. While the senescence is protective, it can paradoxically promote cancer progression in certain contexts. So senescent cells actually secretes a bioactive molecule known as SASP, the senescence-associated secretory phenotype, which includes the chemokines, cytokines, and protease, which further promotes the proliferative signaling, angiogenesis, invasion, metastasis, and immune suppression. Senescence is also associated with significant epigenetic and metabolic changes, and epigenetic modifications such as histone modifications and DNA methylation can drive the transition to the senescent state and influence the SASP. Also, there's a therapeutic implication of senescence. So targeting the senescent cells is a promising therapeutic strategy. So senolytic agents which selectively eliminate senescent cells and xenomorphic drugs which modulate the SAPs are being developed to overcome therapy resistance and improve the cancer outcomes. So basically, cellular senescence is a double-edged sword in cancer biology. While it initially acts as tumor suppressor by arresting the proliferation of the damaged cell, the persistent senescent cells can promote tumor progression through the cell and contribute to therapy resistance. Another thing which they included in the 2011 paper was the drugs. So they didn't mention most of the drugs or most of the hallmarks because they believe that most of these drugs are undergoing trial, and they didn't want to mention those drugs. But a certain group of drugs are actually using this hallmarks ability to target the cancer. So we have different target therapies. We have EGFR inhibitors, for example. We have telomerase inhibitors and PARP inhibitors. Uh, that was the all. My reference was from these three papers, and all the images were from the Robbins Book of Pathology. Thank you.